All right, so let's see what we remember from these three processes. All right, so first, when we're dealing with DNA, let's compare DNA and RNA first. DNA versus RNA. All right, so tell me three differences that you remember about DNA versus RNA. Right, so first, what is DNA, by the way? What does the DNA stands for? Anybody? You can text it or speak aloud. DNA.
Deoxyribonucleic acid for DNA,
Right.
and RNA will be ribonucleic.
That is correct. So we have deoxyribonucleic acid and RNA is just ribonucleic acid. All right, so what are some differences that you know between DNA and RNA?
RNA is single-stranded and DNA is double-stranded.
All right, so this is double-stranded. Single-stranded. Right. Anything else you remember?
So the sugar in DNA is deoxyribose and the sugar in RNA is ribose.
Right. Anything else?
DNA has thymine and RNA has
Right.
uracil.
So in terms of the bases, RNA has uracil and DNA of thymine. Okay. So these are the three important differences between DNA and RNA. In terms of the bases now, there are two P words that we use to describe the bases. So some of the bases have two rings and some only have one ring. Do you know the P words I am referring to? If it has two rings versus one ring?
Sir, the two rings one is purines and
Right.
the one ring would be pyrimidines.
Right. Pyrimidine. And do you know which ones are the pyrimidine?
Sir, the C and the T, the cytosine and the thymine.
Okay. Add the U as well. So I like to use the acronym. So cytosine, uracil, and thymine. And so for the purine, it would be adenine and guanine. So as you said, the purines, there are two rings. And the pyrimidine, a single ring. Now, in terms of the structure of our nucleotide, right? The nucleotide is made up of three things. What are they? The nucleotide has three components.
So the nitrogenous base, the pentose sugar, and phosphate group.
Right. So nitrogenous base, pentose sugar, and which other one? No, sorry, yeah, pentose sugar and the phosphate group. And to represent this now using a diagram, once you see the word pentose, what does that mean? How many carbons are present?
5.
5.
That is correct. And you're only required to use a diagram to represent it. All right, so question, what is the five-sided shape? What is this called? So we have three things: nitrogenous base, pentose sugar, and phosphate group. So this in the middle would be
So the base.
the sugar in the middle here. Right, so this is the pentose sugar. This over here now is the nitrogenous base. And this would be the phosphate group. You can just put a P, but let me just, so this is the phosphate group, right? So this is a diagram. Showing structure a nucleotide. All right, so in terms of the structure of DNA, right, of two types of bonds. Types of bonds, DNA. Anybody want to take a guess? Which type of bond is present in DNA? Answer me one. All right. So we know that.
Peptide.
Please repeat.
Peptide.
Not the peptide. Peptide is for protein.
Sarah phosphodiester bond.
Right. That one. Hydrogen as well. So the two of them are hydrogen bonds. And the next one, phosphodiester bond. Right? And this is a covalent bond. So we have hydrogen bonds and we have covalent bond, specifically called the phosphodiester bond. We won't have to go into the chemistry of it, of how you get the actual ester bond. All right, so the chemistry of it is not required. So where does the hydrogen bond comes into play? The hydrogen bonds are used for base pairing. All right, so you won't find hydrogen bonds on a single strand of DNA. So let us say you have one strand of DNA, A, G, C, E, A, and over, right? So this is one strand of DNA. This bond here, this is not your hydrogen bond. So on a particular strand of DNA, you will have your, I'm just going to put covalent bond, all right? But it's the phosphodiester bond, all right? So on your single strand of DNA, that is where you have the covalent bond, all right? But if you try to form a base pair now, so like A on one strand of DNA would bond with T on the second strand. This now is your hydrogen bond. That means hydrogen bond is used to hold the double helix of DNA together. So we said that DNA is double-stranded. So it is the hydrogen bonds that will hold the two strands of DNA together, because the base pairing is formed through hydrogen bonding, which I do from module 1 at the start of module 1 when you learn about water. You also did hydrogen bonding. On a particular strand of DNA of covalent bonds, and between the two strands now you have hydrogen bonding taking place. Right? So those are the two types of bonds you have in DNA. Right? And the last part of this intro, let's look at the actual base pair. So I already stated that A bonds with T. This is so we know that right. So we know that A bonds with T. Now, do you know how many hydrogen bonds are between A and T? Anybody? Actually two. And by the way, if we are showing hydrogen bonds, we normally use broken lines. Right? For hydrogen bonds, if we are showing it, we use broken lines. Covalent bond, however, it would be a solid line. So between A and T, in terms of hydrogen bonding, hydrogen bonds. Right? However, the GC bond is a triple bond. So each set of broken line represents one hydrogen bond. So GC, it has 3 hydrogen bonds. And AT has 2 hydrogen bonds, which means that GC bonds are harder to break than the AT bonds because it has more hydrogen bonds. Right? So this is what you need to know about in terms of the structure of your nucleotides, the base pairing, and a little difference between DNA and RNA. I'm going to pause here if you need to take off anything. All right, so first thing, before we cut the actual process and the enzymes, DNA replication, it is said to be semi-conservative. But anybody know what the term means if we say semi-conservative? Right, what does the prefix semi mean? In general terms, right, half. And what does, so conservative, conserve, right? So if you're conserving something, what are you doing? What does conserve mean?
Save.
All right. So you are saving half of the information. So when DNA is replicated, half of the information is retained. So let's look at a strand of DNA. So we have two strands of DNA. Just join them. Now, in the process of replication, we know that the two strands must separate. So you will have the black separating from the blue. Now we know that DNA, the strands, they are complementary to each other, right? So meaning if this was an A down here, you would have a T. So you have the blue, we have the blue, right? The black is complementary to the blue. And the blue is complementary to the black. All right. Now, just this was your original DNA, right? Original. And this blue line was also the original as well. So this is the new strand of this DNA molecule. This is the new, that is one DNA molecule, and this is a next one. So the top strand is the new one. So now, in this new DNA molecule, right, it has a new strand, and it has one strand from the original DNA. So this is the original DNA molecule. So these new ones now, one of the strand is new and one is from the original. So one of the two strands is new. So 1 over 2 times 100, that would be 50%, right? So DNA replication, it is semi-conservative because half of the info is being retained. So this from the original DNA molecule, only one strand is being retained. This one is new strand. Okay? So that is why we say it is semi-conservative. Right? All right, so in terms of the process of DNA replication, there are two types of DNA replication. Are not two types, two forms of DNA replication. One is continuous What is the next one?
Discontinuous.
Right. So DNA replication, it can be continuous and it can be discontinuous. So it's not just about memorizing the enzymes and knowing to list the steps. They can just ask you for discontinuous replication, which would be different from explaining continuous replication. If they ask you to explain DNA replication, you will have to explain both continuous and discontinuous. Right. When we speak about continuous replication, right, versus discontinuous, we are referring to a particular strand which is being replicated. So each strand on DNA is given a particular name. Let me see if you remember. Which strand would undergo continuous replication? So put the name of the two strands. You're probably interpreting it incorrectly. So you have the lagging strand, and you have which other strand? There's a lagging strand, there's also a of the leading strand. All right. So the lagging strand, that one is what we refer to as discontinuous replication. And the leading strand is continuous replication. So if you're asked to explain continuous replication, they are telling you to explain how the leading strand is replicated. If they ask you to explain discontinuous replication, they are asking you to explain how the lagging strand is replicated. But if they just ask you to explain DNA replication, then they are telling you to explain how both strands are replicated. Right, so we are going to do both of them now. I'm just seeing your answer, Nadiv. All right, what are the enzymes involved in DNA replication? Can you tell me some names? All right, we know we have helicase, right? Any other enzyme that you can recall? So we have helicase, DNA polymerase 1 and 3, and ligase. Remember, if you can remember this one, but it's not, I won't have to mention it for you. But you must remember helicase, DNA polymerase 1 and 3, and ligase. Also have topo isomerase, right? And this is actually the very, very first enzyme that is used. Even before helicase, of topo isomerase. Third, This is the fourth one. Fourth, one will be the fourth. And this one will be the fifth one. Right? So you must know your enzymes. All right, so let's look back again now at the DNA molecule. It's a double helix, right? The first thing that needs to happen, if you want, the strands has to separate, as you know. The thing about DNA, the double helix is tightly coiled, right? Also, the function of the topoisomerase is to ease the tension. Before helicase come and start to break the hydrogen bonds to separate the molecule, topoisomerase is actually ahead of it. So all of these enzymes are working with each other, like one in front. So just imagine a conveyor belt with topoisomerase ahead, followed by helicase, then you have your DNA polymerase. Left out one enzyme, pyramase, which is to make the primer. Know that the DNA strands, we use 5' and 3' to indicate direction. And we know that the strands run anti-parallel to each other. So this strand is 5', this one here is 3'. So that means this must be 3', and this one here must be 5', right? Now I'm going to start with the leading strand. Which is continuous replication. All right. So we know that topoisomerase will first bind to the DNA, right? To ease the tension in the molecule. Helicase, all right, so just let me draw helicase. Let us say this is helicase, all right? Helicase can move along the molecule. Let me just illustrate the process. Let's just put let's say these are hydrogen bonds, right? Holding the molecule together, right? So the helicase will move along the DNA and break the hydrogen bonds. All right? So If it breaks the hydrogen bonds, then the strands are going to separate. Okay? For explanation purposes, I'm going to draw two straight lines. Now, if I'm breaking the bonds in this direction, so we are going in this direction, right? So that means DNA replication is starting at this point, right? So I'm going to call this our origin of replication. So wherever DNA replication starts, that's the origin of replication, right? So that means replication is starting here and going in this direction, right? We know that the leading strand, what direction does the leading strand run? All right, we're not sure. Let us figure it out. So we have a red strand, right? And we have a blue strand. The red strand here, 5 prime, this is 3 prime. Here is 3 prime, and this is 5 prime. DNA replication, all right, let me leave it. Very simple and straightforward. New DNA is made in a 5 prime to 3 prime direction, right? Now to understand this, I am going to put in the direction of which DNA replication is taking place. So when we say new DNA is made in a 5 prime to 3 prime direction, this is not the full story, right? It has to be in the direction of where replication is taking place. Okay? So question, if this side is the left side and over here is the right side, in our example, is replication taking place from left to right or from right to left? In what direction is DNA replication taking place in our example here? Are you unsure or not? Left to right? No, but thanks for answering. All right, so remember here, this side of the molecule is the origin of replication. So helicase is going to bind here and move along in this direction. It is starting from the right, moving across the DNA to the left-hand side. So DNA replication is moving from right to left. So when I say new DNA is made in a 5 prime to 3 prime direction, right? That means that DNA is being made 5 prime here, 3 prime out here, right? So when you're making DNA, it is made 5 prime to 3 prime in the direction where replication is taking place. So replication is taking place right to left. At the right is a 5 prime, and at the left it will be a 3 prime. So if replication is started based on this diagram, right? It's starting on the right hand side. So that will be a 5 prime. And when it finish out here, you will have the 3 prime. All right, my question for you, do you know which of these strands is the leading strand and which one is the lagging strand? I need some feedback so I know if you understand. So this is your original DNA, and this is the new strand of DNA that you will make. This new strand, it is parallel to this, and it is anti-parallel to this one. Whichever strand is anti-parallel to the new strand, that is your leading strand. So whichever strand is anti-parallel to the new DNA, that is your leading strand. The one that is parallel is the lagging strand. So that means this strand is your leading strand, and this one is your lagging strand. All right? Is that clear? Understand?
Yes, sir.
All right. All right, so that means which strand will be replicated continuously? This one here or up here? Right, so it has to be, please repeat.
Continuous will be the leading strand.
Right, the one up here. All right, so we are going to focus on this strand up here, and then we look at the one down here. I will tell you why it is the leading strand. Right, I'll give you a minute if you want to take off anything off the board, and then we continue. All right, so helicase, it attaches, moves along the DNA. All right, so let's say when it's here, right? All of this portion of DNA from here to here would have been separated, right? We're going to focus on that part. And we said the red one is the leading strand, right? Now, as you know, let us just put on some nucleotides, right? So the nucleotides would be sticking off, right? Sorry, the bases. So to make a new strand of DNA, so let us say you have ATG here. To make a new strand of DNA, DNA polymerase 3 is going to add the complementary base. So if you have A here, you would put down T, then A, and then C. And that is how this new strand will be made, right? And it will just continue along until it is completed. However, DNA polymerase 3 cannot just go ahead and start adding nucleotides. It needs a little, it needs some form of initiation, right? And that what gives it the what we call the initiator, right? It's the RNA primer, right? So an RNA primer is going to be added, all right? After helicase unwinds a portion of the DNA, we're going to an RNA primer. So the primase, the enzyme, they are going to make an RNA primer at the end of the molecule, all right? So this is your RNA primer. Now, once the RNA primer is there, then the DNA polymerase can go ahead and bind. Which color do I want to use? RNA polymerase, not RNA polymerase, DNA polymerase now. It will move along the DNA. So helicase is ahead, right? Helicase is moving along, breaking the hydrogen bonds and separating the DNA strands. DNA polymerase, sorry, just clear that. So DNA polymerase 3 will come along now and move along the DNA strand behind of helicase and just add in the complementary nucleotides. So that is what will happen. So it will just move along the DNA, adding the complementary nucleotide, right? Until it reaches the end, right? Just like that. Now, in terms of 5', 3', remember your original strand up here, the leading strand, it was 5'. This one was 3'. Remember, your new strand is made 5' to 3'. So this is 5' and this is 3'. So notice your DNA, the two strands are anti-parallel to each other. And as we said, it is semi-conservative because this new DNA molecule, one of the strands is from the original DNA. One of the strands is from the original DNA, and one of the strands is new. All right. So let me put this in words. So we know that helicase will bind to the DNA molecule at the origin of replication. It breaks the hydrogen bonds between the base pairs and so the two DNA strands are separated. No, it's when these strands are separated, the hydrogen bonds can actually reform to give you back the double helix. In order to prevent that, single-stranded binding proteins So the single-stranded binding protein, they bind to the DNA strand and prevents it from reforming. So like when you have this open, the proteins will bind to it, and so no hydrogen bonding can take place. Continue now. An RNA primer made by RNA primase as a side note, a primer is a short sequence of nucleotides. Then now DNA polymerase 3 can come into play. And we just add right here an RNA primer. An RNA primer is made at the origin of replication. Made at the origin of replication. Right, and then DNA polymerase 3 binds to the primer. First to add complementary bases. So that is where the ATCG comes into play. It just moves along the DNA, adding the complementary base. If it's a A, it add T. If it's a C, it add G. If it's a G, it add C, vice versa. The DNA polymerase 3 binds to the primer and starts to add complementary bases as it moves along the DNA molecule. All right, we have put bases, even though it's a complementary base, the base, the base is actually attached to a nucleotide. So the, it's not like the base is by itself. The base is a part of the nucleotide. So it is attaching nucleotides. All right, so I'm going to put nucleotides. If you want to use complementary base, you have to mention the word nucleotides, because it will not be 100% accurate to just say base. All right, we can say start to add nucleotides, start to add nucleotides that as the complementary base. All right, so the COM there is short for complementary. So the DNA polymerase, it is going to be adding the nucleotides that has the complementary base on it. All right, so quickly again, with continuous replication, once the strands are separated, once the strands have been separated, an RNA primer is placed, right, where you have the origin of replication. Once you have the primer, DNA polymerase will bind, move along the DNA, and just add the complementary bases, right? And that's it. It will just continue until it reaches the end. Now, once it reaches the end, you cannot have RNA mixed in with your DNA. So DNA polymerase 1 will remove the RNA primer and fill it with DNA, right? So that's it for the continuous replication. It's very straightforward, right? So I will give you a little time again to write this up, and then we look at the login strand. All right, so we are finished with continuous. We're going to do discontinuous. All right, so let us look at why it is discontinuous. Remember, new DNA is made 5' to 3' in the direction that DNA replication is taking place. So new DNA has to be made so this is my new DNA in red. Now, as you can see, the original strand, which we described as the lagging strand, was 3' and 5'. 3' here, 5' here. My new strand is being made 5' to 3'. Remember, DNA replication is taking place in this direction. Good. As you can see, both strands are parallel to each other. But we just said that DNA has to be anti-parallel. So clearly, this can't work. So one of the strands has to go 5' to 3'. All right? One strand, the two of the strands, they cannot be 5' here and 3' here. So one of them has to turn around. It cannot be the original one, right? Because it is already a full completed strand. So the one that can turn around is the one that is just being made. And that is what will happen. It is being made 5 to 3, only that this time it is not going in the direction of replication. It is actually going in the opposite direction. Let me just say this again. New DNA has to be made in a 5' to 3' direction, right? However, in our example, DNA replication was taking place from the right-hand side to the left-hand side. But because your original strand and the new strand would be parallel to each other, we cannot have that taking place. And so the new strand that is being made is still 5' to 3'. However, it is going against the direction in which replication is taking place. Now, because it is going against the direction in which replication is taking place, there are going to be some issues that needs to be sorted out. And for that reason, it will not occur as smoothly as with the leading strand. So it will not be a continuous process because the new DNA that is being made is going against the direction in which replication is taking place. It will not be continuous, all right? So now let's look at why that is. Let me just move this, move this, move this. Now remember, DNA is like this, right? Wind up, pile up. Good. So when helicase comes along and it opens a portion of, let me use a different thing. So helicase will come along, right? And open up a portion of the DNA. So helicase will open up a portion of the DNA. Now let us say that this one down here is our leading strand. You put on your primer, right? Just going to put P for primer. And then your DNA polymerase right, will come along and just move along, making the new strand. When helicase reaches out here, DNA polymerase will be right behind of it to keep on extending the strand. Right, so helic polymerase, it will just be right behind it to keep adding. Now, forget about that strand. Remember we said no, that the new strand, it has to turn around. What that means, you cannot have a primer here. So we are looking at the lagging strand now. You cannot have a primer here, because if you put the primer here, here will be 5 prime, good, and out here will be 3 prime, which is in this direction. So for that reason, you have to put the primer further down on the DNA that has opened up. And so wherever you have the primer, that is your 5 prime end. Now remember, here hasn't opened up as yet. This is the part that has opened up. And so the new DNA is going to be made for this segment. All right? So this is the 5 prime end and this is the 3 prime end. Now, when more DNA has opened up, what is going to happen? When more DNA has opened up, just ignore the leading strand. We will have to put now a new primer. All right? This is a new primer because your DNA polymerase would have completed this segment and reached the end of the DNA, and that's it for that segment. However, the process is not complete because now you have a next portion of DNA to fill out. Let me just move helicase. And so this segment will be filled out by DNA polymerase again. And so by that time, by this is to be completed, more of your DNA will be opened up. And so you will need a next primer because remember, DNA polymerase cannot start until there is a primer. So you put a primer here, DNA polymerase comes and it makes this segment and it's done. But then you put a new primer here, and so DNA polymerase makes a next set of DNA. Give me just a second. Yeah, so you make a new primer again. Yeah. And this portion of DNA, it will be made again. All right. So as you can see, once you have this primer, it starts and it's done, right? And then you have to start over again and start over again. So it's a process of starting and stopping and starting again. Right? Question, do you understand this? You want me to do it again or you're good? Let me know. All right? Take it that you understand. So that is what will happen on this strand. So you will not have a primer at the end. Go over. All right. So we are trying to make a new strand now from this DNA. All right. However, I cannot put a primer here. Wherever I put the primer, that's the 5' end. If I put the primer here, that will be the 5' end for my new DNA, right? So if we take it as is, when the new DNA is made, it will be parallel to each other, all right? And that cannot work. It has to be anti-parallel. Because we need it to be anti-parallel, what has to happen is that you put the primer further down on the DNA. Good. Remember here, let me just put closed. Okay? So this portion of your DNA, it hasn't been opened up as yet. It is closed. So your DNA polymerase, this is the 5' end, will come and add the complementary nucleotide. All right? So there you have it, it is anti-parallel. Now, when the DNA polymerase 3 has completed this segment of DNA, the portion that was closed will now be opened. And so a new primer will be made, right? And let me put back closed. So let's say this portion hasn't been opened as yet, only this part. So once a new primer has been made, DNA polymerase can attach itself again and complete it. All right, and that is what it will do. Attach itself and add the nucleotides until it is completed. Just erase this a little. When you see me join the lines, right? I am actually representing base pairs. So DNA, again, this is the primer. DNA polymerase will come, will add T, will add G, will add A, and T. And so this portion of the DNA has been replicated. And then some more of it would have been opened up and say I make a new primer. Let's say in this portion, you have G, G, A, E. DNA polymerase will come again. Where it's G, it will put C, C, P, A, G. Right? Nothing will come here because that is where your primer is. Right? And so this portion of the DNA has been made again. And it will continue like that until it reaches the end of the DNA. So let me just put a little more out here to complete the process. Let's say I have A, P, and C, right? Of course, you would have to have a primer, okay? Primer. Again, polymerase will come, so this would be T, E, A, and G, right? That part, primer, right? And the primer there. Good. And so we have completed the replication of the DNA molecule. Now, clearly, this is not one continuous thing. This is, let's call it segment one, segment or fragments. So this is fragment one, fragment two, and this is fragment three, right? We have a special term for these fragments. They are called Okazaki fragments. Once you hear the term Okazaki fragments, that is referring to the lagging strand and discontinuous replication. Now, do you see why it is discontinuous? Is it any clearer? Okay, good. All right. So let's put our 5 prime here. Up here was 3 prime. So that is how we manage to ensure that the two strands are anti-parallel. So the reason why right. So the reason for this whole starting, stopping, and flipping around the how the strand is made, all because it has to be anti-parallel. All right. So the only thing that needs to happen now is that DNA polymerase 1 will come in, remove the primer, right? And fill it with DNA. All right. Just going to put this called here the DNA. Right? Fill the parts with the primer with DNA. And then DNA ligase will join up these fragments to give one complete DNA molecule. Right? I'm going to write this on the board now in steps.